Hello everyone, my name is Mustafa and I sincerely thank the organizers of 6th Annual Congress on Plant Science and Biosecurity for this wonderful opportunity. So today my topic of presentation is eco-friendly seed priming approaches for breaking dormancy in Albizia julibrissin. We call this tree as Persian silk tree in common language. Also, this research was conducted at Comenius University, Bratislava under the supervision of Dr. Bojana Shara. I also want to say thank to the funders. It was funded by EU Next Generation and EU through the Recovery and Resilience Plan for Slovakia. Let's move to the next slide. So, before diving into the main topic, let me introduce this tree, commonly known as Persian silk tree. This species is remarkable for its multiple uses. It provides timber for construction, fiber for clothing, and raw material for furniture making. Also, it serves as a fodder and forage for the livestock, offers protection from floods in hurricane-prone forests. Also, it has significant medicinal value. Additionally, it is widely used and appreciated for its ornamental beauty and plays important ecological role through nitrogen fixation. So basically, it is very important tree in different aspects of the life. Furthermore, this tree species was selected for this research because of it belongs from the Fabaceae family, and most of the Fabaceae family members show dormancy, either physical or morphological dormancy. So it can act as a simulation for breaking seed dormancy. Let's move to the next slide. Let me explain the core problem we addressed in this study. Albizia seeds have hard, impermeable seed coat that causes physical dormancy, preventing water uptake and germination. As you can see in this figure, if seed faces unfavorable external conditions, seed tends not to germinate. They refuse to germinate. So we address this problem. Also, in a recent context, or nowadays, which is very hot topic, that is climate change. Changing temperature make unfavorable conditions to the species to grow. So we selected this species of Fabaceae family, that how we can break seed dormancy in order to overcome this dormancy and low germination problem. In next slide, I'm going to tell how we investigated this problem. We investigated 3 distinct pathways to break physical dormancy. Pathway 1 is mechanical scarification, where we clip the seed tip coat to create a physical opening which allows water to enter and initiate germination. We achieved almost 99% of germination. In second pathway is hot water treatment at 80 degrees Celsius, which softens the waxy layer and disrupt the palisade cells, increasing permeability, resulting in 96 to 97% of germination. Pathway 3 is our novel approach using non-thermal plasma, which employs reactive species UV and ions to interact with seed surface, achieving approximately 90% of germination. Next up, in this experiment, I'm going to explain the experimental design in the slide. We tested 8 different methods to break physical dormancy. We named them as alphabetically in alphabetical order, like A, B, C, D, E, F, G, and so on. Treatment A was mechanical scarification using scissors. Treatment B and C involved hot immersion at 80 Celsius for 1 minute and 2 hours respectively. Treatment D was simple water soaking for 2 hours, and treatment E combined water soaking with a 7-day drying period. Treatment Treatments F and G used cold temperature at -18 degrees for 1 hour and 16 hours. Finally, the H treatment was thermal plasma with corona discharge at 9 kilowatts and 3 microamps with exposure times ranging from 1.25 minutes to 40 minutes. So these were the treatments we used in this experiment. Let's move to the next slide. Here I want to explain that how non-thermal plasma was incorporated for breaking seed dormancy. The system consists of high voltage source, if you look into the picture, which generates almost 9 kilowatts at 300 microamperes. A microjet needle positioned at 3 millimeters above the distilled water creates the plasma discharge. The seeds are placed in the water below the needle and the platinum wire serve as a ground electrode. We tested 6 different exposure times which range from 1.25 minutes to 40 minutes. Let's move to the next slide. So these are our final germination percentages. After 7 days, the control group showed only 10% germination, which is very low under unfavorable conditions. Among the NTP treatments, we observed the clear dose-dependent response. Treatment 1 or T1, which consists of 1.25, T2 showed minimal effect at 12% and 25%, while T5 reached 48% and T10 reached at 60% and T20 reached 70%. So you can see that the T40 was the best treatment in plasma regime, non-thermal plasma regime, which scored almost 90% of the germination. So the traditional methods, they perform best. Mechanical scarification achieved 99% and hot water for 1 minute reached 96%, and hot water for 2 hours achieved 97% germination. And the question here you might be thinking about that, if the traditional methods are breaking dormancy effectively, why don't we use the traditional method and we go for plasma methods, non-thermal plasma methods? Basically, this is a simulation project, and non-thermal plasma can be incorporated into the different members of these species. So we used this non-thermal plasma just to test that it can be used as a model testing or model dormancy breaking methods for other species as well. Let's move to the further results. This time series graph shows the germination dynamics over 7 days. Scarification showed the earliest onset with 28% of germination by day 2, reaching 99% by day 7. Hot water treatment for 1 minute had delayed start with 0% until day 3, but then rapidly rose to the 96%. Hot water for 2 hours shows medium early germination and steadily progress to 97% at this day 7. And treatment D through G, they showed minimal response, remaining below 10% throughout the entire period. Let's move to the next result. So these graphs illustrate how non-thermal plasma affects seed surface properties. On the left, you can see the contact angle decreases with increasing plasma exposure time from 99 degrees at 0 minutes down to 77.7 degrees at 40 minutes. The critical transition from hydrophobic to hydrophilic occurs between 5 and 10 minutes, crossing the 90-degree threshold. On the right, you can see imbibition percentage increases proportionally from 0.08% in the control to 1.85% at 40 minutes. This directly links surface modification to improved water uptake and faster germination. Let's move to the next result. So The non-thermal plasma treatment also enhances seedling vigor. We measured 3 vigor indices. Vigor index 1 based on the seedling length multiplied with germination percentage shows progressive enhancement for T2.5 onwards, reaching maximum at T40. Vigor index 2 using fresh weight followed a similar pattern. Vigor index 3 based on dry weight also showed dose dependent improvement. All three indices confirm that non-thermal plasma not only improves germination but also produces more vigorous seedlings with better biomass accumulation. Let's move to the next slide. So this is comparative metrics. Here, scarification achieves the highest germination rate at 99% and is fastest to germinate, but causes seed coat damage. And this is kind of labor intensive work. Hot water achieves 96 to 97% germination with minimal damage and is simple to scale but carries a bunch of risk. Non-thermal plasma achieves 90% with no seed coat damage. It has enhanced post-germination vigor and high operator safety as well. For small nurseries, in this research, we recommend the scarification or hot water treatment. For large-scale industrial proportions, NTP offers advantages in automation and quality. Let's move to the next slide. Here, if we compare the pathways, all the pathways effectively overcome the water impermeable barrier. Mechanical scarification provides a quick physical solution with 99% success, while hot water treatment offers thermal alternative achieving 96 to 97% germination. All methods are eco-friendly. I want to emphasize on this point very much because they are low cost, scalable for nurseries and afforestation programs. Also, I want to add few words about non-thermal plasma. Non-thermal plasma is kind of gaining importance in agricultures and botanical works due to its low impacts on environment. There are no leachates to the environment if this non-thermal plasma is applied directly to the seeds or either on the seeds or in the form of spray. So this could be a novel approach to break seed dormancy, increase germination, and also protecting the environment. If you go to the traditional methods, they are equally effective, but they contain few risks. For example, the hot water treatment or things like that. So the key insights is that once the hard seed coat is breached, water can penetrate, which triggers a cascade of metabolic processes that lead to successful germination and seedling establishment. Let's move to the next. To summarize, I want to summarize this thing. Scarification at 90, which gives 99% of germination. Hot water for 2 hours gives 97% of germination, and hot water for 1 minute gives 96% germination. All of these germination results are more than 95%. And also the non-thermal plasma at 40 minutes achieves approximately 90% germination, which represents a novel and non-destructive alternative with enhanced seedling vigor. All methods are eco-friendly in this research with no chemical, they are low cost, safe, and scalable. For future work, we need to optimize the NTP dose response curve. We need to investigate molecular mechanism which involves ABA and GA hormones, scale to continuous flow plasma system, test on the other Fabaceae species, and conduct an economic analysis that which method is more economically low cost and also good for the environment. So these are the key takeaways and the future directions as well. Furthermore, again, I want to thank the funding agency, which is EU, Next Generation EU, through the Recovery and Resilience Plan for Slovakia. And now, if you have some questions, you can email those questions, because I believe this presentation, I'm recording this presentation, so I might not take the questions directly, but you can send your questions at my email, which is ghulam1@uniba.sk. Again, once again, thank all of you for your attention, and for future collaboration, if someone is working on seed simulation, seed dormancy, seed priming, I am available to collaborate in future. Thank you so much for your attention. Have a nice conference and have a nice day. Thank you so much.